I just had a slide I wanted to share, and I was just thinking of like, what would Google tell me, what is a phenotype, and kind of how have we been thinking about this as a foundation? And so I put a couple of a slide together. Honestly, it's really simple just to kick this off. I'm just trying to share my screen. Hold on one second. Just to give a quick broad overview. Let's see if I can present this. Why isn't it giving me presentation mode? Am I not hitting something there? Okay. So can you all see this one slide? Yeah.
Yep.
Okay, great. So we started it back in 2019 just to show that, we've been thinking about this for several years now and started collecting some data, which has many different components of what data could be. But it was very much the start of our longitudinal natural history study. So we looked backwards about retrospectively what the patient's journey was like. And we had a team that published on that genotype phenotype paper by Dr. Wendy Chung and her team at Columbia back in 2019. And that recently got published in 2021. And since then, we've been kind of contributing different components of data. So I just wanted to share this with the group here in terms of all the different bubbles of types of data that Project 8P is looking at. And so I can just start on the bottom left here quickly, but a lot of what you're contributing in the registry and what you did with Dr. Chung is some of your patient-reported surveys. You shared your genetic lab results, possibly contributed biospecimens to rerun lab results and found new findings, and may have been asked questions about, as a caregiver, what does this look like for you? And how does this impact you? And then may have shared some of your MRIs and different imaging and files and clinical reports with our team here, or either with Dr. Chung, and we've linked that. So you'll see all these little link symbols, but we're basically connecting all of those different sets of data to try to make sense of that. We also had, and Dr. Demarest will talk about some clinician reported observations and scales, and that's really kind of the focus of today too. Possibly connecting that to some of your electronic medical records or electronic health records, and being able to do that in an efficient way. It's not easy, but we're starting to be able to connect some of this. And there's a lot of international governance challenges there too. And we're trying to do our best here, but we really need everyone to contribute based on what our researchers and clinical team would like to see to be able to help us answer questions. There's obviously published data on different 8P genes, and collectively, there's also unpublished data, that we may not know about, but we were calling different scientists that may have studied this but didn't report the data. Maybe it wasn't statistically relevant yet. Maybe they didn't have the time to continue working on whatever gene in the 8P region they might have been studying. So, there's an effort here. We have a biorepository that some of you have contributed samples to and specimens, and we're going to start running some lab tests on those. And also yourselves probably have some blood. I'm just trying to be as simple as possible here, but some laboratory results, maybe you ran some tests on your 8P hero that could be interesting and give us some information, insights on what we're seeing in the disease. Some of our researchers are here on this call and, not only reporting results on what they're observing, but also maybe negative results that they can't necessarily publish, but could share with us here in this team science platform. We're also going to start thinking about launching an easy way to see and visually see data like videos or through photos, what we can observe over time of your 8P hero, and be able to share that back with researchers and clinicians. And then if on the bottom right, and this is not, in any order of importance necessarily, it's just collective, is having a lot of data that breaks it down at the transcriptomics level and genomic level, RNA level. And so that's a project that's partially being sponsored by Illumina to do that for those that contributed biospecimens. So this is kind of the big macro lens of what we're trying to achieve here. And we're very much in the early stages of this, but I just wanted to kind of start this conversation off with that big picture. And without further ado, I'd like to introduce Dr. Scott Demarest, who is not only the co-clinical director of the multidisciplinary clinic for neurogenetics that we launched last year, but has also been very much a friend to our community. And I think that is so important. You know, I'm sure everyone here can relate that when you have a doctor who is not only knowledgeable broadly about rare diseases that have neurological phenotypes and symptoms, but that actually can just be such an easy phone call away when we have pretty frightening situations that happen because of the disease and what happens to our loved ones. And so I feel humbled and to have him on our side, and just so grateful again. So Dr. Demarest, you can take it away and share the screen.
Thanks, Bina, and thanks for the kind introduction. I'm the one who feels honored to be able to be part of this amazing community and all of your lives. And that was a really fun way to start, despite you being humble about it, Bina. It was a fun way to start to talk about phenotypes. And I didn't say anything because I had a feeling I'd get the last word. And I agree with everyone. You know, phenotypes are all of those things. It's a layered concept, and it's sort of how deep are you zooming in some ways when you're thinking about what is the phenotype for a particular thing. And as a clinician, we're often asked, what is a phenotype that's related to the particular etiology or cause of that disease? And what's just that person and unique to them? And maybe they got it from other aspects of their family or their environment or any number of other factors. And that's not always totally straightforward to piece apart and tease out, but I'll give you a little bit of a sense of what we're trying to do in our clinic and the types of data that we're collecting. This is pretty high level. We're not zooming in super deep today. It's more of an overview. But the goal is to be able to say, these are really interesting things. We want to zoom in and we want to get really detailed phenotyping here. And so we're open to people commenting and saying, hey, no, I think this is really important. We want you to spend more time on this or that. And please speak up as we go along. Happy to have this be somewhat interactive. All right, so that's who I am, and I already was introduced, so thank you. And this is a picture of our hospital. This is a multidisciplinary clinic. I've listed here some of the members of our team, and the next slide is going to be dedicated to that. And it's important to sort of note, if not already aware, this is nested within a sort of an overarching neurogenetics clinic that is a collaboration with 3 other rare disease groups as well, S6BP1, Ring14, and SLC6A1. And partly that's because there is at least some overlapping phenotype, which means that there's common things that we can work towards in terms of both data collection and clinical care. And it helps with rare disease to be able to do that sort of collaborative work where there's overlapping goals and needs. This is what our team looks like. So 11 providers see a patient on a particular day. We go in groups and work together on that to try to not make things too overlapping or redundant. Dr. Margarita Saenz up here at the top is my co-medical director for the clinic as a pediatric geneticist. Tristan Dinkel over here is our nurse coordinator. She's the one who she runs the thing. She makes it happen. Patients get to the right place, get into clinic, everything due to her amazing efforts. We have a group of special care clinic providers. These are pediatricians who specialize in the care of patients with more complicated disease. So while they're general pediatricians, they're incredibly good at handling genetic conditions of a variety of types. Dr. Mann is our rehab specialist and physical medicine specialist. We have a palliative care specialist dedicated to the clinic who doesn't see every patient, but sees some of them, and that's usually not part of that day, but available when necessary to help families to think about prioritizing decisions and what is leading to highest quality of life. It's palliative care, which can be triggering for some people, but really we think of it as quality of life clinic. Dr. Mealy is our neuropsychologist, so she does all of our psychoeducational testing and helps to clarify strengths and weaknesses for particular individuals and how to adjust educational plans to meet their needs. And I'll show some data from her as well. And then we have our incredible team of therapists, occupational, physical, and speech, and Andrea, Dana, and Drew, who dig into everything about how an individual moves from a gross motor, fine motor perspective and communicates. We've got a team of genetic counselors involved. One of these pictures needs to be updated with someone else who's also part of the team now, which is Katie Angioni. So I apologize to her that she's not accurately represented here, but she's actually our primary genetic counselor for the clinic. We've got social work represented. And our most recent addition to the team is Dr. Ann Reynolds, who's one of our developmental pediatricians, who helps a lot with behavioral challenges and things like that, and is a godsend in my opinion. So just an overview of what we've done so far in terms of 8P in our clinic. So 15 patients have been seen in the last year or so. 12 are on the waiting list currently. 8P patients who have reached out want to come be seen in clinic, and we are working on getting them in. We have plans in 2023 to expand the number of clinics we do per month. So hopefully we'll be able to get not only all 12 of those patients in, but hopefully even more than that in 2023. Our geneticist, Dr. Sines, knows of 10 additional patients who have been seen at Children's Hospital Colorado who are not yet plugged into our clinic. And we're working on trying to connect with those families and see if they would be interested in coming to the clinic. I think it's important when we're thinking about this to just preface that our primary objective for our clinic is to provide the highest quality clinical care. We are trying to make sure that there is comprehensive and very well-coordinated care being given to the families who come to our clinic. That's the primary thing that we're trying to make sure that patients and families get out of that clinic. But it also gives us an opportunity to try to collect data that we can utilize to learn about the variability, the phenotype of 8P. And I like to think about phenotype longitudinally, which we sometimes call natural history studies in the clinical world. And that's really just bringing the idea of phenotypes not static, but ever changing. And when we talk about that clinically, we tend to use the word natural history study. And we'd like to be able to move in that direction with the clinic to be able to generate data that gives us a picture of what does 8P look like, not just in terms of variability at this moment, but over the life course. Of those 15 patients we've seen, 60% were female. The range in age was 5 years to 45 years old. And they've come from all across the country, 7 different states, more clustered around us, but also wide ranging. So what are some of our initial insights from the first year of experience? And I think it's important to realize this is our 1 year of experience. Most of our providers had not seen large numbers of 8P patients before we set up this clinic. So we are really growing in our experience as well. And these are some of our initial thoughts, but there's still more for us to learn as well. And that might influence the types of data that we want to try to capture and our sort of hypotheses about what's important in this disorder or disorders, if we want to think about it that way, since it's really a group of different types of genetic changes. 73% of our patients had some evidence of unique facial features, smaller size of the mouth, prominent forehead, wider set eyes, and arched palate. 93% carried a diagnosis of intellectual disability of some form or fashion. 73% had the changes on the brain MRI that we have known about previously, where there are abnormalities of the corpus callosum. 40% have had cardiac abnormalities, and 40% have had seizures, of which half were currently requiring seizure medications. This is something I think is probably a bit unique compared to—
Sorry, can I ask a question?
Yeah, please.
Sorry. For the remaining 27% that do not have corpus callosum abnormality,
yeah,
can you identify the genetic variation in these individuals? And are these individuals the same as the 27% that don't have any facial anomalies?
Yeah, great questions. Not yet would be my answer for that. These are relatively small numbers still. I think those are the types of questions we'd love to be able to ask. So cross-referencing brain abnormalities with facial feature abnormalities, or with deletion or duplication size, or other things like that, to try to understand what's going on and what's the mechanism here, similar to what's been done with GATA4 for cardiac disease, and I'll touch on that in a minute. We have not yet done those associations, and I think sometimes when we have small numbers, we need to be a little bit careful about spurious associations, but I think that's exactly the kind of question we'd like to ask.
So for these 15 patients, do you actually know the size of the duplication, deletion, and all these, I guess, genetic changes? So that is known.
Yes, we have their genetic, clinical genetic test reports. And so we're able to sort of map to those. I will say, depending on the technology used, because these are clinical tests, sometimes there are different levels of clarity of exactly where, breakpoints might be and exact sizes of them, just depending on the technology that was used to make that clinical diagnosis. Many of these patients have also been offered to be involved in the other data sets that Bina alluded to at the beginning. And so to the extent that they may also end up in a biomarker repository or included in the Illumina sequencing projects, there may be additional, more detailed sources of genomic data that could also be brought to bear. And we're happy to always work with Project AP to crosswalk and make sure that we can utilize that data fully.
Yeah, I think even for some of the things that we would like to do, I think having this divergent set of samples would be very beneficial. This is great.
Yeah. So cortical visual impairment, and I'm going to start with just defining this, is where there is a dysfunction of visual processing rather than something that's abnormal with the eye. So the eyes are fine. They send signals back to the brain, but the brain maybe doesn't process those as effectively as other individuals, and it can lead to some impairment in the way in which individuals interact with their environment from a visual standpoint. This is a highly underdiagnosed challenge across medicine. So that's not unique to 8P or other disorders. It's sort of caught between two specialties. Ophthalmologists say, the eyes look fine, and neurologists say, I don't know, that's a vision thing, and neither wants to make a diagnosis. We see it across a wide range of neurogenetic conditions and are pretty well tuned into it in Colorado. And so it's something that I've been assessing for in all of the patients that we see. And we've actually seen it in 2/3 of patients, some level of cortical visual impairment. It's also not the same as blindness. There can be degrees of severity associated with it, which I think is really important. If we think about visual processing from a neurophysiology perspective, it is incredibly complicated. You have certain parts of your brain that are dedicated to seeing movement in particular directions, or certain colors, or certain contrasts. And an individual may have some impairment in some set of those challenges, but not other areas. And so this can be a really interesting thing to try to characterize. And I think one element to this is about 1/4 of your brain is actually dedicated to visual processing. So this is an important aspect of brain function. Faye.
I just asked, so presumably a child could have glasses and still have this going on, right? They could still have some form of visual impairment, but also have the processing errors.
Absolutely. And so there may be certain challenges that an individual has in terms of eye alignment or refractive errors, which is a eyeball issue, which an ophthalmologist or optometrist will diagnose and give glasses for potentially, or other things like that. But just because they have that, which is, again, quite common, many of us have to wear glasses, that doesn't mean that there's not also a brain thing. So CVI is specifically pointing towards the brain side of visual processing, and that may be in combination with needing glasses or without having any sort of ocular issues at all. Alice? Sorry, you're muted.
So presumably, like, even though your sample size of patients is 15, because it is 10 out of 15 with evidence of this, that does feel quite sort of significant to you, even though it's not a massive sample.
I think that this is going to be an important phenotypic feature as our numbers grow. I don't know if it's going to be 2/3. There certainly could be some bias in the 15 patients that we've managed to connect with so far. And maybe it's half or 1/3, or maybe it's higher. I don't know. That requires us getting to a bigger sample, but I think there's going to be a proportion of patients where this is an issue.
And the second part of the question is, this is the sort of thing where coming to the clinic really helps with the diagnosis. Like this, you know, there's a lot of information parents contribute, but this is one where there's a real, like, benefit of them being seen in your clinic.
I think so. Because again, I think this is something that is often very much underdiagnosed. I think even if you don't come to our clinic, or that's not logistically feasible, I know we have a global community here, raising the question of this with local providers is also probably reasonable. And sometimes people don't make the diagnosis because they don't feel confident about it, or they just don't think to. And so I think it would be a reasonable thing to do locally as well.
Thank you. That's really interesting. Thank you.
Was there another question?
All right, I'm just going to jump in. Who would you have make the diagnosis?
Whoever's comfortable.
Okay.
Sometimes it's an ophthalmologist, sometimes it's a neurologist. And the reality is that from a neurology perspective, you want to have had an eye exam by a good, ideally, if you're a pediatric patient, pediatric ophthalmologist, not optometrist,
yeah.
MD, and have them look for any pathology or any challenges, problems with the eye itself. If they're not seeing any issues there, and there is functional evidence by the way someone interacts with their environment that there may be some vision challenges, that's really what gets you to a CVI diagnosis. Unfortunately, many ophthalmologists don't feel confident to make that diagnosis by themselves, and neurologists don't feel confident to make that diagnosis without you having seen an ophthalmologist. I also feel that way, but most of the patients we've seen have actually seen an ophthalmologist at some point who said, yep, the eyes are okay. And so then I feel comfortable saying, you know, this is what I think is going on.
Sure, more clear cut. Thank you.
Can I ask a very...
Good.
Can I ask just a very basic definitional question? Sorry. When you say cortical vision impairment, are you talking about cerebral cortex vision impairment or non-ocular vision impairment? So basically, is this including problems with motor control derived from the cerebrum, or is this truly cerebral cortex vision impairment in these individuals?
So this is something that's probably not as well understood as we would like. I will say that there are some cultural differences. In the US, we tend to use the word cortical visual impairment. In Europe, they tend to use the word cerebral visual impairment. I don't have a strong feeling that one is necessarily better than the other one. Generally speaking, we are talking about non-ocular visual impairment. And so that may be that it is an element of disruption of visual pathways behind the eye. Usually we can see that on MRI if that's really what's going on, like an optic nerve abnormality, for example. and that's not what we tend to be seeing in this population. My gut is that this really is a brain function issue, a cortical issue. And now, where in this complicated process, I think is something that needs further elucidation. So it could be that there's challenges with the cortex processing that information primarily. There could be visual association cortices that are disrupted or dysfunctional, or it could be, as you're sort of alluding to, connections between the visual cortices and motor cortices that coordinate tracking and eye movement and things like that. We have another term, oculomotor apraxia, that often, but not always, can accompany cortical visual impairment and may involve very obvious motor abnormalities of the eye, but that's often in conjunction with some element of CVI. I hope that answers your question.
Yeah, I think it's. I'm very excited that you're discussing this. You know, we have a number, as you know, we're part of the challenges we're working with is understanding gene relevance through all the different phenotypic elements, and there, in terms of gene expression, we have a number of genes that are expressed in the cerebral cortex, but then also a number of genes expressed, you know, with high enrichment in the cerebellum, which would be motor control, right? And so understanding whether it's a motor control issue versus an issue in vision processing, I think, is important distinction.
Yeah, I think that makes a lot of sense. And Travis, we have specific elements of our exam that we do capture that may point a little bit more towards cerebellar involvement or not. Part of what I'll say is, in order to coordinate those movements, you have to have visual processing that starts. And so if you have, we know this from neurology, so like if you've had a stroke that's taken out your visual cortex, you'll develop abnormal eye movements. And that's just a lack of input into the pathways that then would control the eyes, rather than a primary cerebellar or eye tracking field abnormality or something along those lines. But it's also possible that you have some amount of visual input that is getting in and getting through, but that there is a concomitant cerebellar or movement abnormality that is confounding the situation. So it may be a bit complicated to tease apart, but we do have some specific data points that might help us. There one other question? Sorry, yeah, go ahead.
Yes, this is Jean-Pierre Zadid speaking. Is there, for children above 4 years old that you've seen, have you done an fMRI on them to actually identify clearly, you know, and I'm talking about strictly any of the children that do not have, or any adult patients that do not have any sort of deformities that could impact their eyesight. So we're talking about strictly high integrity structures, and then we're going through an fMRI like a normal individual to figure out how the blood flow works, what are the cognition capabilities, and whether they're seeing in, you know, up to the optical chiasma if anything is disturbed there.
Yeah, so the short answer to that is no.
Okay.
There are some issues of sort of what are validated clinical tests that would make sense and be worth you know, the expense of doing that outside of a research protocol. I think it's an interesting idea, and if you have other thoughts, I'm happy to connect about what we could do and look for and things.
Thank you.
I'll skip sort of going through bullet by bullet on this, but this has important clinical implications as well as the research implications, which we've discussed some. And were there any other questions before I move on? All right. Another aspect is communication. This is looking at it more from a clinical side of things. When we think about the utilization of augmentative and alternative communication devices, or AAC devices, for individuals with any sort of communication challenges. we found that approximately half of our patients had not previously tried any sort of assistive communication device prior to coming to our clinic, and that in essentially all of them, we had made a recommendation to consider the use of one, or at least a trial. This can have a pretty big impact in terms of the ability to communicate both at home and at school, to have access to those educational learnings for individuals who are not able to communicate effectively verbally, but may still be able to communicate wants, needs, and associations through a device. And then I think what we would say just clinically is while we do sort of a basic assessment for this as part of our multidisciplinary clinic, for those who are local or logistically there are the opportunities for this, our speech therapists will do a deeper dive and actually test devices and work on this over time for patients where that logistically makes sense to do that. Where it doesn't, our speech pathologist has engaged with local speech pathologists for families to help them sort of explore that and give their thoughts as well. Now we're getting back to the genetic findings we asked a little bit in terms of before. This is just sort of broad categorization of the patients that we have. 2/3 have been in the inversion del dupe category, and you can see that there's been a range in terms of the size of duplication and presumably deletions as well. But this data is available to kind of do deeper dives with. Pure deletions, 4 out of 15, and 1 that was a pure duplication. This known association with GATA4 and congenital heart anomalies, we did see that 2 patients had that had deletions in GATA4 did have congenital heart anomalies. It's also worth noting that we had 6 patients who had congenital heart anomalies, and so there are patients who did not have a GATA4 deletion who still had cardiac anomalies. And then just sort of making a plug that from a clinical perspective, genetic counseling is provided with our clinic for families to help them understand the implications of this. And ideally over time, we all better understand the implications of the specifics of a particular genetic change and what that means in terms of what's happening with an individual or what phenotypic elements that explains, and potentially in the future, what do we need to watch out for, or what should we expect in terms of life course based on those genetics. That's a tall order. That's going to happen over time in bits and pieces, but that's where we certainly want to go with this. Bless you.
Sorry.
It's okay. So neuropsychological evaluation. Of our 15 patients, 14 were able to complete comprehensive neuropsychological evaluations. This is a pretty intensive process, usually lasts a couple of hours with Andrea Amelia, our neuropsychologist, to probe various different types of neurologic function and quantify that neurological function using specific tools. That's sort of the process for neuropsychological evaluations. Of those that had this, 9 were in the inversion del dupe and ranged from 4 to 13 years, dupe only at the age of 9, and then distal deletions, 4 of them ranging from 6 to 27 years. So this is, again, relatively small data. This is somewhat labor intensive for families and investigators to get this type of data, but I think it's a gold mine when you can have it. And so I'm really excited that we're generating this as part of our clinic. We utilize this data to help us to be able to provide guidance back to families about how to approach individuals from an educational standpoint, but it also gives us some really rich data so that we can think about neurologic function and the impact of 8P changes for individuals. Some of our basic associations, some of our basic associations that we're seeing this way are that, generally speaking, the more severe end involved inversion del dupe patients in terms of what was their age equivalency. But I think we have to be really careful about this because this is a single point in time and we have a range of ages. And so part of the question that we'd like to get to, and this is where we move to a natural history type set of data set, is what's this look like over time? How does this compare when you're 4 years versus 8 years versus 15 years old? and the trajectory that an individual can go through in terms of those strengths and challenges from a psychoeducational perspective. Again, sort of broad strokes, we saw that motor skills tend to be a little bit better developed earlier on compared to language skills. We have some sort of age equivalencies that we can think about in terms of how do we scope this and what do we tie this back to. I think there's limitations to thinking about it from that perspective, but it is one of the ways we can quantify this. There was one 8P duplication patient, dupe only, that had average cognitive function, but still had a number of behavioral difficulties. I think that's an interesting thing to sort of, as our patient population grow in our data set, we can think about different aspects of cognitive function versus behavioral challenges and where they might be coming from a genetic perspective. And then neurocognitive functioning, sort of for those with the distal deletion, tended to be quite variable. And so it is an interesting thing for us to, again, be able to try to expand on over time as we get a larger data set. Any questions there? Heidi.
Is there an ideal minimum age that is, as far as what you can offer to families and data that you can get from families? Is there an ideal age to make the trek for a clinic visit?
Yeah, I think that's an important question. So the way our clinic is set up, we try to do this neuropsychological testing the day prior to the big clinic day, so we can still have families who are traveling make a single trip, but it becomes a 2-day visit instead of just a 1-day visit. For local families or ones able to travel, we can split those up and have people seen separately. So that's just a logistical element to this. We can gather data about development at any age, including infants. The tools we have available when you're under 2 years of age and then sort of under 3 or 4 years of age are different. So what tools might be available or useful to try to capture that information does change as you get older. It also changes based on functional ability. What we can utilize to capture those with more significant challenges compared to more mild challenges is also different. I think by the time you're into toddler years, and for many individuals already involved in therapy or have an IEP or something along those lines, then you want to start to be thinking about some of these elements. Certainly by the time you're in school age, so 5 years and older, we want to make sure that there is an educational plan that's appropriate to that individual. Many schools will, and they are actually required by law in the United States, to perform testing and cater to the needs of that individual. Different schools do it better than other schools. And so I would sort of throw out that from a clinical perspective, anytime that there is challenges or you feel like the school's just not hitting the mark, that's a great time. We're happy to help and intervene and be an advocate with you for that. In terms of when can this type of testing be as clinically helpful, I think at the younger ages, it may not be as useful to that family. It is useful for us from a research perspective because it does help us to try to picture the full age course a little bit more as we can capture at least some information even in those early ages. Hope that answered your question. Faye, go ahead.
Hi, can I ask how consistent do you find the age grouping? Because I presume you break the test down into, different sections which should address different sort of cognitive functions. So if, they test sort of like it's a 10-year-old who tests it, a 5-year-old capability in one section, do they also then test as like an 8-year-old in another section? You know what I mean?
Yeah. So I think that's actually a really insightful question, Faye. And that is one of the things that we would like to delve into deeper. So our general experience with neurogenetic conditions is that an individual doesn't always follow across different domains at the same rate. And so while they may be making developmental progress, we all know that these individuals are making progress on their own timeline and in their own way. And not only do they not always make progress at the same rate in different domains, but they don't always follow the same developmental order that we expect for typically developing individuals. So they may have developed the ability to achieve certain tasks that are generally considered more advanced or more complicated, but not have achieved certain things that seem more basic. And that happens in these populations. And so being able to not only get bigger data sets, but data sets where we have multiple time points in the same individuals, I think is really helpful for us being able to understand, is it just, well, language comes in a little bit later, but then it catches up and ends up at a similar age equivalency by the time you get to 10 years of age? Or is language always at a lower age equivalency compared to motor, for example? Is that genomics dependent on what type and size and location of the genetic changes? Those are all really nice questions that we want to be able to ask as we get a better data set. It's a little bit hard to do with relatively small numbers, but I think those are the types of things that are generating certain hypotheses for us at this stage. Like, oh, I wonder if there's a connection between this and this, but we may not be able to fully answer it yet.
Sure. I'd love to get more data about like what's involved with this test to see if we could, have it done here in the UK. I mean, it would be invaluable. I mean, I'd pay anything for this, for us to have this, to give to the school,
Yeah. There are certain tests that are relatively easy to administer. An example of that would be the Vineland is a one test that would be utilized in this space.
Okay.
And I think Bina could talk a little bit more about different potential data registries and sources that we're trying to collect Vinelands for. That only sort of looks at it from one perspective, and it can actually be done just as a parent survey. It has some limitations when done that way compared to being administered, but don't let perfect be the enemy of good, right? We can collect data and get there. The type of testing I'm alluding to here is these are PhD trained individuals who then do additional training in clinical neuropsychology. It's a pretty advanced skill set that Dr. Mealy is bringing to the table to do this. I know that there are individuals in the UK who have those same skill sets. I don't know how available they are or what their time looks like. And it's generally something that has a lot more demand than there are individuals to do it.
Sure. Well, if there's any way you can share the details with Bina, that'd be brilliant. Thank you.
Yeah, absolutely. And we could easily produce a sort of rundown of what are the different types of tests that have been performed. And actually, Bina, this is included in our abstract that AES is sort of a breakdown of the different types of tests that have been utilized and what those scores look like.
Thank you.
Yeah, sure, we'll share them. Oh, and the Vineland 3, the parent-reported Vineland 3 will be available to all AP families in January. It's, it's useful, but it's not always, it's not a perfect standardized measure either. So we've had over 50 families complete them in the past and gleaned some insights from it. But we will launch it again along with a couple of other standardized surveys. And then I'll share more about what is more clinician administered, possibly like when we huddled again as families.
And I think Bina, Andrea would be interested in thinking about, like, is there certain subsets of the Vineland that might be useful to look at with bigger numbers based on some of the things that she's seeing with the in-person testing, right? And so I think she'd be happy to try to help dig into that and see if there's some other things that we can get out of that data.
Okay, thank you.
This is sort of, I'm getting towards the end here, but I've alluded to future directions on a number of occasions already, and this is sort of summarizing that. Really, the point is to be able to get to better understandings of genotype-phenotype correlation. I'll throw out there that may not just be DNA genotype, that may be functional impacts of DNA rearrangements and other things that we have to get to be able to tease these things apart. We'd like to be able to continue to make both a larger data set, but also make sure that we're getting a sufficiently robust data set in terms of the depth that we can provide to be able to make these associations. And then we'd love to at some point be able to get this towards a clinical care guideline. There's sort of different levels of care guidelines. There's sort of a, hey, you should pay attention to these things kind of care guideline, which I think actually we're starting to move in that direction from Wendy's paper and the type of data that we're collecting. I don't think we're that far away from that. Being able to say like, oh, you should do this treatment or that treatment requires a different level of quality of data to be able to get there. But I think that comes over time. And so we certainly want to help to move things in that direction. And that's the end of what I have. Any other questions as we finish up?
Hi, I have a question.
Yeah.
So first, thanks very much. This was a really informative presentation and the very different perspective that I think those of us on the research side are used to. So thank you for that. I'm wondering if I imagine you don't have enough data right now, but I'm wondering if eventually you see being able to sort of classify patients into sort of, there's really, this is not one disorder, but 3 disorders. And of course, each one has variability, but there's some discrete grouping, possibly related to genotypes or something like that. Because I think for those of us who are making cellular or tissue scale models of these kinds of things, having the idea of like, well, how many models do we need to capture the disease, and can we see correlations between what we see in these tissue models and phenotypes? It would be very useful to think about that kind of thing.
Yeah, I totally agree. From my perspective, a little bit of the challenge is I don't honestly know now. Are we talking about 3 different categories, or are we talking about a more continuous continuum of abnormalities and changes? And, when you have deletions, duplications, translocations, rearrange, and you have varying sizes, I think it becomes, there's so many variables associated with that, that trying to think about how do these group can be challenging. And I don't personally have my head around it right now. I'm open to ideas. And if you guys tell me like, oh, I think that these are the things that group, I'm happy to look and say, do we see clinical grouping in those as well? So I think some of it is we're still in the hypothesis phase from that perspective, I think personally, but I'm excited to continue having that conversation. I'd love to get there as well.
Yeah, I mean, I think you actually have many, you don't have a lot of data, but you have many more, like we currently have 1 IPS line from a patient. Hopefully we'll have another couple in the next couple of months. But I mean, even forgetting about the genetics, do you think there's a point where you can say, well, there's some phenotypic grouping where, I mean, just imagining like putting all your data into some big quantified data set and running dimensionality reduction things on it, or like, is there any sense in which that would be useful?
Maybe. A lot of times being able to do sort of guided cluster analyses or other sort of AI-based stuff are going to be more successful when you have larger data sets than when you have smaller data sets. I certainly see certain clusters, but to me, it's like a bunch of overlapping Venn diagrams rather than sort of more discrete clustering of things. But I think that's something we're still also trying to get our heads around. Like, do we see CVI more often when we see X neurocognitive profile? I think there's going to be some associations there, but I don't want to speak prematurely about them as well. But I think that's something we're trying to get at.
Okay, thanks.
Nicoletta.
Hello, hi. Thank you very much for your talk. Indeed, we have a very different perspective after this. And I was just wondering whether you would be able to link some of the clinical phenotypes to other diseases that have a single gene dysfunction, perhaps to localize a bit more the link, the cause, the genotype with phenotype as we discussed. And I was thinking about you mentioned prominent forehead. Is there anything related in other diseases where there is an impairment in apoptosis, for example, in development, that would link. So this type of link, which I know it's hard to do with 15 patients, but I don't know if you have anything on that or you plan to.
So part of the challenge is that any of the features that I mentioned are represented and seen in numerous other disorders, not sort of just a few. They tend to be quite common. There's sort of an element that clinically there's common final pathways, particularly within neurologic diseases. We have thousands of genes associated with intellectual disability and developmental delay. We have about 1,000 genes associated with a risk for seizures. We're starting to increase our knowledge that visual impairment is quite common among neurogenetic disorders. It's been present in at least more than half of the patients that I've looked for it in, for example. So it tends to be a bit challenging to sort of aim for trying to zero in from that perspective, because we end up getting common clinical presentations from a diverse set of etiologies. The same thing is true even for the facial features. Those are quite common, and we see them across a large range of disorders. We see them a little bit more represented for overarching chromosomal abnormalities compared to single gene disorders, but that's not an absolute rule. And it isn't isolated to only certain chromosomes. It can be lots of different chromosomal abnormalities.
Thank you.
Yeah. Janie, sorry if I mispronounced anyone's name.
Oh, that's fine. First of all, as an AP mom, I just want to say it was a great presentation. I'm wondering, do you still only see US families at the clinic, or do you have European families as well?
We are open to trying to see more families from outside the US as well. The biggest challenge ends up being a practical one of our insurance system in the US. And we do bill for seeing patients and families clinically. And so that this many providers, it ends up being a pretty hefty bill. And so I think if it's not covered by insurance, that can be a bit of a barrier. That's not to say we're not interested in it and happy to do it, but that's sort of a logistical complication of it.
Okay.
I'm always happy to be connected with any local providers if they're just like, hey, have you seen this? Or do you have any thoughts on this? I'm happy to do that sort of informally as a consultation to another provider that you have, even if you haven't been seen in our clinic. There was another hand up, I think, a minute ago, but I lost it.
Stefan.
Hi, yeah, that was me. Great talk. I'm not sure my question is a good question, so I'm tempted not to ask it, but since you called on me, I hope you didn't cover this, and in some ways your point about insurance actually does answer it. But working on iPSC models, so basically Aria's question made me think about this. So working on iPSC models, we like to have an isogenic model, right? And of course you can't have this in the clinic, but you do have in most cases the carrier with you, right? At least the carrier of the inversion. Do you think this might be a really silly question, but do you think there would be any diagnostic value in taking the parent through the same battery of phenotyping to establish sort of a way of controlling for the rest of the genotype? So to basically clean the scores and get a genotype corrected score for the patient.
So not a silly question at all. Great question. I'll say we do not universally see the inversion, for example, from mom. So certain aspects of this can be seen in the parents as a setup to this, and other aspects seem to be completely de novo. And so there is some variability there, but that's not to say that there wouldn't be some value when it's present to look at that. The other challenge is that whether we see it in the parents as germline, or if it's something that is actually just in germ cell, in eggs or sperm or something like that. Right? And so we don't always have access to knowing the case, and that obviously would have implications about whether we're seeing that phenotypically in the parents. But I think it is an interesting question. There are some additional logistical things to being able to start to do this, like neuropsychological testing, for example, in parents, which could be interesting. We also have a lot of other things that make us who we are by the time we're adults and can cloud the picture a bit. And we have to have separate IRB sort of approvals to make sure we can do all those. So there's some logistical things there, but it's not a bad idea. And there are definitely chromosomal disorders that are, there are other examples of where parents have certain issues that are different than what children have. And there's a known association that way. MECP2 duplication disorder is an example I'm thinking of where we can see that type of phenomenon between mothers and their children.
Great, thank you.
The one other thought that I had that I would just throw out for our research community, particularly if you're doing animal-based phenotyping, there's very frequent conversations about what translates between animal models and humans, and how do we think about correlates. So for CVI, for example, I know groups for other disorders where there's CVI, where they've done visual evoked potentials that you can do in mice and rats and things like that, and do see that translates. And so there may be things, and I'm happy to have conversations about that as well, where there might be an interest in sort of saying what battery of phenotyping should we be trying to do in animal models and things like that, if that's useful. Things don't ever translate perfectly, but I'm always happy to at least have the conversation.
Thank you so much, Scott. This is great. And it is the last one for 2022 for us. Since then we're rolling into December, and I'm sure none of you want to hop on for a Zoom roundtable. So we will resume in 2023. This isn't a goodbye. I think we're still in touch with each of you, and in different capacities. Katie Syverson did spend quite a bit of time to show us some highlights. So similar to how Scott shared some statistics on observations of those that are seen in the clinic, we also have some from those that have completed the registry. And that's always ongoing. So thank you for those that have consented. And there's quite a bit of information that we're starting to see that is interesting to us, and we'll be sharing with Dr. Demarest's team in the clinic and researchers as well. And they're kind of nicely formatted in Excel spreadsheets too for researchers. So that's all available if you are interested in that data. And as well as families, we're really trying to get a better understanding of what we're seeing based on different age groups and different genetic categories or subsets of chromosome 8p. So we're excited to show that. Sorry, we did not have time, but I actually feel like it's so nice that Scott, this really encouraged a lot of conversation. So I'd rather have it that way. And, we can kind of postpone our talk about the registry insights, and it'll just be really honestly continual of what you presented. So again, thanks so much. Whitney, are there any other household items? Okay.
Other announcements. Just a big thank you to everyone for participating. To date, you've made this a really, I think, successful series.
Yeah, and we're not going anywhere. We're going to continue this. So, and refine it and just get, more on board. I know there's a few researchers that were out today too. So the recording will be shared on our YouTube channel and on social media. So for those that missed it, they can also watch it again. Again, have a wonderful rest of your day. Good night.
Thanks, everybody.
Thank you, Scott. Thank you, everyone.